This is a continuation of the lecture from Unit 4. The genotype is the set of genes in an organism, but not all of these genes are expressed. The phenotype is the physical features or traits of the organisms. Transcription is the process of synthesizing an RNA copy of a gene from the DNA. Translation is the process of synthesizing polypeptides or proteins from the RNA copy. There are a few forms of RNA involved in protein synthesis. The mRNA or messenger RNA will carry the genetic information from the chromosomes to the ribosomes. The rRNA or ribosomal RNA combines with the ribosomal peptides to form the structure of the ribosome. The tRNA or transfer RNA delivers the amino acids to the ribosomes. Transcription begins with the initiation phase. The enzyme RNA polymerase will move along the DNA helix until it reaches a promoter. This allows the RNA polymerase to unzip or unwind the DNA in the promoter region. The promoter is near the beginning of the gene and initiates transcription. Not all RNA polymerases bind all promoters equally strong, which affects the amount and type of protein transcribed. In bacteria, the sigma factor is a portion of the RNA polymerase needed for recognition of the promoter. Eukaryotes use proteinaceous transcription factors to help bind RNA to the primer. The elongation process begins 10 nucleotides away from the promoter. While transcribing the first 10 ribonucleotides, the RNA polymerase releases its sigma factor, allowing it to tightly adhere to the DNA. Many RNA polymerases can concurrently transcribe the same gene. There are some differences in RNA polymerase used in transcription and DNA polymerase used in replication. The RNA polymerase unwinds the DNA itself and does not require the helicase. It is also slower, working at about 50 nucleotides per second. It will incorporate ribonucleotides instead of deoxyribonucleotides and uracil instead of thymine. It also lacks proofreading, leaving errors every 10,000 nucleotides. Termination is the phase when the RNA polymerase and the transcribed RNA are released. Self-termination uses a terminator sequence. The RNA polymerase transcribes a terminator sequence rich in G and C bases, followed by a region rich in A and U. Transcription allows the GC region because there are 3 hydrogen bonds between guanine and cytosine instead of the 2 in adenine and uracil. This will actually slow down the transcription. This allows the formation of a stem and loop structure to put tension on the structure and then pull off the adenine-rich area. In enzyme-dependent termination, it depends on a termination protein that binds to a specific sequence near the end of the RNA transcript. The protein moves towards the 3' end and forces the RNA polymerase and DNA apart. We have a brief transcription animation here. This is produced by the University of Nebraska. Protein synthesis begins inside the nucleus of a cell. Inside the nucleus, chromosomes are stored, Chromosomes contain the information to produce every protein the organism will need encoded in DNA. The information is encoded in the sequence of nucleotides of the DNA double helix. The process of protein synthesis occurs in two steps. During the first step, called transcription, the mRNA copy of the gene is made which carries the encoded information into the cytoplasm. Every gene has three regions that occur in the following order. The first is the promoter, which turns the gene on and off. The second region is the coding region, which encodes the information for which protein to produce. The third region is the termination sequence, which signals the end of the gene. RNA polymerase molecules are present in the nucleus. This enzyme is responsible for reading the DNA code and building the mRNA strand. The promoter region of the gene acts as a light switch. It signals when to turn the gene on and off. The RNA polymerase molecule recognizes that the promoter is signaling to produce the protein. The RNA polymerase molecule binds to the DNA strand, and the DNA double helix unwinds. Once the RNA polymerase reaches the coding region, it reads a single strand. As it does this, it builds the mRNA copy. As RNA polymerase reads each nucleotide, it brings in the complementary nucleotide and bonds them together, forming the mRNA strand. The nucleotides break free from the DNA, and the DNA folds back into a double helix. When the RNA polymerase reaches the termination sequence, the sequence causes the mRNA to fold back on itself. This prevents transcription from continuing, and both the RNA polymerase and mRNA strand fall off the DNA strand. We also have a second animation here of transcription.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates, and the newly formed strand of RNA is released.
In translation, ribosomes read the genetic information and use the code to synthesize a polypeptide. The genetic code is nearly universal with few exceptions. It is a redundant code with more than one codon associated with each amino acid. The codons are triplets of mRNA nucleotides that are associated with a particular amino acid. AUG is the start codon, which codes for methionine in eukaryotes, and N-formyl methionine in prokaryotes, mitochondria, and chloroplasts. The stop sequences are UAA, UAG, and UGA. The mRNA carries an AUG start codon, and then the sequential codons occur after and end with at least one stop codon. Prokaryotic ribosomes can start translation before transcription is done because both occur in the cytoplasm as there is no nucleus. There are some differences in prokaryotes and eukaryotes here as well. The eukaryotic mRNA contains some non-coding regions and is considered pre-mRNA. The non-coding regions or intervening sequences are known as introns, which are removed to make a functional mRNA. The exons are the expressed or coding regions. Eukaryotic mRNA carries instructions for only one polypeptide, where there may be more than one in prokaryotes. The eukaryotic mRNA is not translated until it is fully transcribed because it must move out of the nucleus. The tRNA is made from about 75 ribonucleotides and forms three main hairpin loops. It has an acceptor site for a specific amino acid at the 3' end and an anticodon in the bottom loop. The anticodon is the triplet at the bottom of the tRNA that will pair to a codon. Although there are 64 codons, some organisms will have fewer tRNAs due to wobble in the third nucleotide. The wobble allows a nucleotide other than the normal one to bond by changing the angle. Frequently changing the third nucleotide will not change the amino acid. The ribosome is 70S in prokaryotes and 80S in eukaryotes. Each is composed of two subunits with a smaller subunit shaped to accommodate 3 codons at one time. The A site accommodates the tRNA delivering the amino acid. I remember it as the accepting site. The P site holds a tRNA and a growing polypeptide. The E site is where the tRNA will exit from the ribosome. The subunits of the ribosomes are made of smaller subunits themselves. The prokaryotes have a 50S and 30S subunit. The 50S subunit is composed of a 23S and a 5S rRNA and 34 polypeptides. The 30S is composed of a 16S rRNA and 21 ribosomal polypeptides. In eukaryotes, the 80S ribosome is composed of a 40S and 60S subunit. They have a larger rRNA and more polypeptides than those in prokaryotes. Here we have an animation of translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Here's another short animation on translation. There's another animation here that you can watch on your own on translation from McGraw-Hill. This can either be used from the link in this document, or you may also link to it directly off of the Angel website. Initiation is the first stage of translation. Here, the mRNA, 2 ribosomal subunits, and tRNA form an initiation complex. The smaller subunit attaches to the mRNA with the start codon at the P site. The tRNA attaches to the start codon at the P site. The energy for this binding is provided by GTP. Next, the larger ribosome attaches to form a complex. During elongation, the tRNA with the matching codon attaches to the A site. An elongation factor protein moves the escort tRNA with the GTP, and the energy from the GTP is used to stabilize the tRNA in the A site. The larger subunit, Ribosome, forms a peptide bond between the terminal amino acid and the introduced amino acid, and the polypeptide attaches to the tRNA at the A site. Using the energy from the GTP, the ribosome moves down one codon and transfers the tRNA to the adjacent site. The ribosome releases the empty tRNA from the E site to be recharged, and the cycle repeats itself. This allows another ribosome to follow as it elongates. With termination, the release factor halts elongation. The ribosome recognizes the stop codon and modifies the large subunit. This will activate the ribosome to sever the polypeptide from the final tRNA and dissociates the subunits. Transcription is under strict control. About 75% of the genes are expressed all the time, while others are regulated to produce or synthesize polypeptides only when they are needed.